हेलो गाइस। तो आज का चैप्टर है बायोमोलिक्यूल्स। ये बायोलॉजी का चैप्टर है बायोमोलिक्यूल्स। तो जितने भी क्वेश्चन डिस्कस किया जाएगा सब प्रीवियस ईयर में आया हुआ क्वेश्चन है, सब डिफरेंट एग्जाम में आया हुआ क्वेश्चन है। तो मैं मेंशन नहीं किया कौन सा क्वेश्चन कौन सा एग्जाम में आया है। तो शुरू करते हैं फर्स्ट क्वेश्चन से। इन लेकोपेरॉन द नंबर ऑफ स्ट्रक्चरल जीन्स आर 5, 4, 3, 2. Option C is correct, 3. In Lycopteron, the number of structural genes are 3. 3 is leg Z, and leg Y, and leg A. ये 3 है. In Lycopteron, the number of structural genes are 3. One is leg Z, one is दूसरा is leg Y, and तीसरा is leg A. Next. DNA fragments can be joined together by using. Option is restriction, endonuclease, Okazaki fragments, DNA ligase, DNA helicase. Option C is correct. DNA fragments can be joined together by using DNA ligase. Next question. Nobel Prize to Kornberg and Ochoa was given by artificial synthesis of genes, artificial synthesis of DNA, one gene, one enzyme hypothesis, chemical of DNA and RNA. Option B is correct. Artificial synthesis of DNA. Nobel Prize to Kornberg and Ochoa was given for Kornberg and Ochoa को नोबेल प्राइज मिला था आर्टिफिशियल सिंथेसिस ऑफ डीएनए की वजह से. नेक्स्ट. The fact that a purine base always pairs through hydrogen bonds with pyrimidine base leads to in the DNA double helix. Option is the semi-conservative nature, uniform width throughout DNA, uniform length in all DNA, the anti-parallel nature. Option B is correct. The uniform width throughout DNA. The fact that a purine base always pairs through hydrogen bonds with a pyrimidine base leads to in the DNA double helix. Option B is correct. Uniform width throughout DNA. Next. Question 5. Nucleotides are formed of. Option is purine, pyrimidine bases and phosphates only. Option B, purine, sugar and phosphates only. Option C, purine, pyrimidine base, sugar and phosphates. Option D, pyrimidine, sugar and phosphates only. Option C is correct. Nucleotides are formed of purine, pyrimidine base, sugar and phosphates only. Nucleotides are formed of purine, pyrimidine base, sugar and phosphates. Next. The polypeptide chain is initiated by methionine, leucine, lysine, glycine. Option A is correct. Polypeptide chain is initiated by methionine. Option A is correct. इसके लिए next. Which of the following should be most abundant in animal cell? mRNA, tRNA, miRNA, and rRNA, microRNA, ribosomal rRNA. Option D is correct. Which of the following should be the most abundant in animal cell? Option D is correct. rRNA. rRNA जो है ये forms core of ribosome. rRNA forms core of ribosome and इसका वर्क जो है also essential for protein synthesis. Next. Degeneration of genetic code is due to third member of codons, first member of codons, second member of codons, entire codons. Option A is correct. Degeneration of genetic code is due to third member of codons. Next. Length of DNA in E. coli is option is 5386 nucleotides, 48502 base pairs, 4.6 into 106 base pair, 3 into 3 into 109 base pair. Option C is correct. Length of DNA in E. coli is 4.6 into 106 base pair. Next. Which enzyme is used in the unwinding of DNA? Which enzyme is used in the unwinding DNA? Ligase, topoisomers, helicase, exonuclease. Option C is correct. Helicase. Which enzyme is used in unwinding of DNA? Option C is correct. Helicase. हेलिकेज जो है डीएनए रेप्लिकेशन के लिए एसेंशियल एंजाइम है, डीएनए रेप्लिकेशन के लिए एसेंशियल एंजाइम है हेलिकेज। नेक्स्ट, व्हिच ऑफ द फॉलोइंग ग्रुप्स कंसिस्ट ऑफ पॉलीसेकेराइड्स ओनली? ऑप्शन इज सुक्रोज, ग्लूकोज एंड फ्रक्टोज, maltose, lactose and fructose, glucose, sucrose and maltose, glucose, cellulose and starch. Option D is correct. Which of the following groups consist of polysaccharides only? Option D is correct. Glucose, cellulose and starch. ग्लूकोज जो है ये स्टोर करता है ग्लूकोज को इन एनिमल्स, एंड सेलुलोज जो है ये स्ट्रक्चरल कंपोनेंट इन प्लांट सेल वॉल, एंड स्टार्च जो है ये ग्लूकोज को स्टोर करता है इन प्लांट्स में। नेक्स्ट, the enzymes involved in feedback inhibition are called allosteric enzyme, holoenzyme, apoenzyme, coenzyme. Option A is correct. The enzymes involved in feedback inhibition are called allosteric enzyme. Next, excessive use of which vitamin causes liver damage? Excessive use of which vitamin causes liver damage? Option is vitamin B2, B1, B3, K. Option C is correct. Vitamin B3. Vitamin B3 niacin. Excessive use of which vitamin causes liver damage? Vitamin B3 niacin. Next, the scientific name for vitamin K is ascorbic acid and phytonadione, and tocopherol pantothenic acid. Option B is correct. Scientific name of vitamin K is phytonadione. Option B is correct. Next, during protein synthesis, AUG functions as the initiator codon in mRNA. AUG जो है ये स्टार्ट कोडन है, स्टार्ट कोडन। AUG functions as the initiator codon in mRNA. What should be the tRNA molecule that picks up and brings the amino acid specified by this codon? Option D is correct. USC. Next, in the genetic code dictionary, how many codons are used to code for all the 20 essential amino acids? Option is 64, 20, 60, 61. In the genetic code dictionary, how many codons are used to code for all the 20 essential amino acids? Option D is correct. 61. Next, which side of tRNA molecule hydrogen binds to a mRNA molecule? Option is codon, anticodon, 5' end of tRNA molecule, 3' end of a tRNA molecule. Option B is correct. Which site of a tRNA molecule hydrogen binds to a mRNA molecule? Option B is correct. Anticodon. Next, according to Wobble concept, the G base in the anticodon can recognize the following bases in the codon: U or C, T or C, C only, all the given option. Option A is correct. U or C. According to Wobble concept, the G base in the anticodon can recognize the following bases in the codon is U or C. Next, the number of types of DNA nucleotides depend on the number of types of DNA nucleotides depend on the number of nitrogenous bases, hydrogen compounds, RBC count, WBC count. Option A is correct. Nitrogenous bases. Nitrogenous bases of DNA is adenine, guanine, cytosine, thymine. Nitrogenous bases of DNA is adenine, guanine, cytosine, thymine. Next, the genetic code was discovered by Nirenberg and Holle, Nirenberg and Mathey. Option B is correct. इसके लिए Nirenberg and Mathey. Genetic code was discovered by Nirenberg and Mathey. Next, with which bond the heavy and light chains of the antibodies are linked together? Van der Waals bond, disulfide bonds, ionic bonds, hydrogen bonds. Option B is correct. Disulfide bonds. With which bond the heavy and light chains of the antibodies are linked together? Option B is correct. Disulfide bonds. Next, enzymes that converts all alcohol enzyme that converts alcohol into acetaldehyde in the liver. Option is maltase, alcohol dehydrogenase, acetal dehydrogenase, and amylase. Option B is correct. Enzyme that converts alcohol into acetaldehyde in the liver is alcohol dehydrogenase. Option B is correct. Next, Okazaki is known for his contribution to the understanding of which one of the following? Mutation, transcription, DNA replication, translation. Option C is correct. DNA replication. Okazaki is known for his contribution to the understanding of which one of the following? Option C is correct. DNA replication. Next, which one of the following statement is true as per Chargaff's rule? Option is A equal to C, A plus G equal to T plus C, G equal to T, A plus T, G plus C and equal to 1. तो अकॉर्डिंग टू चारगाफ्स रूल जो है ये इसका स्टेटमेंट है अमाउंट ऑफ एडेनिन is equal to the amount of thymine and the amount of guanine is equal to the amount of cytosine. Option B is correct इसके लिए. Next, which one of the following is the initiation codon for the synthesis of protein in eukaryotes? Initiation codon GUG, AUG. Option C is correct. Start codon methionine. Option C is correct. Initiation codon. Next, which of the following vitamins is soluble in fat? Vitamin B, C, A, B12. Option C is correct. Fat soluble vitamin is vitamin A, D, E and K, and water soluble vitamin is vitamin B complex and C. Next, which enzyme synthesis Okazaki fragments in eukaryotic cell? Option is DNA polymerase alpha, DNA polymerase delta, DNA primase, DNA ligase. Option B is correct. Which enzyme synthesizes Okazaki fragments in eukaryotic cell? Option B is correct. DNA polymerase delta. Next, which of the following are stop codons? कौन सा स्टॉप कोडन है दिया हुआ है? तो ऑप्शन बी इज करेक्ट. UAA, UAG, UGA, ये तीनों स्टॉप कोडन है. एंड स्टॉप कोडन का जो फंक्शन है ये क्या करता है? ट्रांसलेशन को टर्मिनेट करता है. एंड स्टार्ट कोडन जो है ये है AUG. AUG इज ए स्टार्ट कोडन एंड इसका और एक नाम है मिथियोनिन. स्टार्ट कोडन का जो फंक्शन है ये ट्रांसलेशन को इनिशिएट करता है. एंड स्टॉप कोडन जो है इसका फंक्शन है ये ट्रांसलेशन को टर्मिनेट करता है. नेक्स्ट, DNA sequence that changes its location on the genome is termed as plasmid, cloning vector, transposomes, expression vector. DNA sequence that changes its location on the genome is termed as, option C is correct, transposons. Next, which of the following is not an essential component of protein synthesis? Option is ribosome, oxysome, amino acid, mRNA. Option B is correct, oxysome. Which of the following is not an essential component of protein synthesis? Ribosome, amino acid, mRNA, all are proteins or are essential component of protein synthesis. Oxysome जो है ये ATP को synthesis करता है, adenosine triphosphate को synthesis करता है. Next, Z-DNA is characterized by absence of guanine base, left-handed helix, 12 base pair, turn of helix. Option D, distance between two bases 3.4 A. Z-DNA is characterized by, option B and C दोनों correct है इसके लिए. Z-DNA is characterized by left-handed helix, 12 base pairs per turn of helix. Next, which of the following is not an mRNA codon? यूरासिल है, ये भी यूरासिल है. ऑप्शन सी इज करेक्ट. थाइमिन, एडेनिन, साइटोसिन ये DNA का है, तो ऑप्शन सी इज करेक्ट. नेक्स्ट, which of the following amino acid is coded by initiation codon? इनिशिएशन कोडन AUG, जो स्टार्ट कोडन है, स्टार्ट कोडन AUG मिथ्योनिन। मिथ्योनिन का फंक्शन जो है ये ट्रांसलेशन को इनिशिएट करता है एंड स्टॉप कोडन का फंक्शन है ट्रांसलेशन को टर्मिनेट करता है। ऑप्शन सी इज करेक्ट। क्वेश्चन इज द नंबर ऑफ न्यूक्लियोटाइड पेयर्स, नंबर ऑफ न्यूक्लियोटाइड पेयर्स इन ए complete pitch of Z-DNA 10, 11, 12, 12.5. Option C is correct. The number of nucleotide pairs in a complete pitch of Z-DNA is 12. Next, which enzyme works in conjunction with SSB protein? Which enzyme works in conjunction with SSB proteins during DNA replication? Question is, which enzyme works in conjunction with SSB proteins during DNA replication? Option is helicase, DNA polymerase, RNA polymerase, ligase. Option A is correct, helicase. SSB का जो है which enzyme works in conjunction with SSB means single stranded binding proteins during DNA replication. Option A is correct, helicase. Next, chromatin is chemically made of. Option is nucleic acid, histone and non-histone protein. Option A is correct. Chromatin is chemically made of nucleic acid, histone and non-histone protein. Next, question 37. Enzymes can stop the reaction, change the reaction, catalyze the reaction, none of the above. Option C is correct. Enzymes can catalyze the reaction. Next, which of the following polypeptide chains has the correct sequence of amino acid? Option is glycine, methionine. Which of the following polypeptide chain has the correct sequence of amino acid? Glycine, methionine, lysine, valine. B, lysine, valine, methionine, glycine. C, valine, lysine, glycine, methionine. Option D is correct. Methionine, glycine, valine and lysine. Next, polypeptide chain formation in protein synthesis is initiated by. Polypeptide chain formation in protein synthesis is initiated by aspartic acid, glutamic acid, alanine, methionine. Option D is correct. Polypeptide chain formation in protein synthesis is initiated by methionine. Next, replication of DNA occurs in S phase, G1 phase, G2 phase, M phase. Option A is correct, S phase. Replication of DNA occurs in S phase. Question 41. A gene of 600 nucleotides will code for a polypeptide chain of dash amino acid. A gene of 600 nucleotides will code for a polypeptide chain of dash amino acid. Option is 200, 202, 300, 198. तो इसका जो अगर कैलकुलेट किया जाए, each codon, हर सिंगल codon जो each codon is made up of 3 nucleotide. तो codon for one amino acid equal to each codon made up of 3 nucleotides. तो codons for one amino acid equal to 600 divided by 3 equal to 200. Option A is correct इसके लिए. Next, which one of the following nitrogen base is present in DNA but not in RNA? Cytosine, uracil, guanine, thymine. Option D is correct, thymine. Present in DNA but not in RNA. Option D is correct. uracil तो RNA में प्रेजेंट रहता है. तो क्वेश्चन ने बोला DNA में प्रेजेंट रहता है लेकिन RNA में प्रेजेंट नहीं रहता है. ऑप्शन डी इज करेक्ट, थाइमिन. नेक्स्ट, द मिनिमम लेंथ ऑफ सिस्टोन in base pair which synthesizes a polypeptide chain of 100 amino acid is 100, 200, 300, 400. The minimum length of cystine in base pair which synthesizes a polypeptide chain of 100 amino acid is, तो इसका जो है each codon made up of 3 nucleotides. तो यह होगा 100 into 3 equal to 300. The minimum length of cistern means gene, in base pair which synthesizes a polypeptide chain of 100 amino acid. So each codon made up of 3 nucleotides. So 100 into 3 equal to 300. Option C is correct. Next, ascorbic acid is also known as vitamin B, vitamin D, vitamin C, vitamin A. Option C is correct. Vitamin C, ascorbic acid. Next, name the vitamin formed by carotene, which is a vegetable pigment. Vitamin B, D, C, A. Option D is correct. Name the vitamin formed from carotene, which is a vegetable pigment. Option D is correct. Vitamin A. Next, during mRNA synthesis, the unwanted DNA regions are removed and amino acid coding regions are joined by the process of capping, termination, splicing, methylation. Option C is correct. Splicing. During mRNA synthesis, the unwanted DNA regions are removed and during mRNA synthesis, the unwanted DNA regions are removed and amino acid coding regions are joined by the process of splicing. Option C is correct. Next, enzymes are biocatalysts. Enzymes are biocatalysts and they catalyze biochemical reaction. Enzymes are biocatalysts and they catalyze biochemical reaction. Which of the following reaction is not catalyzed by an enzyme? Unwinding of 2 strands of DNA, dissolution of CO2 in water, formation of peptide bond, dehydrolysis of sucrose. Option B is correct. Dissolution of CO2 in water. Next, which types of rRNA are found in 80S ribosome? 80S राइबोसोम कैसे हुआ है? जो लार्ज जो है लार्ज 60S एंड स्मॉल 40S, ये टुगेदर फॉर्म्ड 80S राइबोसोम. टुगेदर फॉर्म हुआ है 80S राइबोसोम. लार्ज राइबोसोम जो है वो कंटेन करता है 28S, 5.8S एंड 5S एंड 5S RNA, and the small ribosome contain small 40S ribosome contain only 18S. तो इसका जो करेक्ट ऑप्शन है ऑप्शन ए इज करेक्ट. 28S, 5.8S, 5S एंड 18S. ऑप्शन ए इज करेक्ट इसके लिए. नेक्स्ट, which of the following is a stop codon? AUG, GUG, UU, UAA. Option D is correct. Stop codon is UAA. And stop codon UAA और भी है UAA, UAG and UGA. AUG जो है म्यूथेनिन, ये स्टार्ट कोडन है AUG. नेक्स्ट, which of the following does not take a part of DNA replication? DNA replication में कौन सा पार्ट नहीं लेता है? लाइगेज, हेलिकेज, DNA ऑक्सीडेज, RNA पॉलीमरेज. ऑप्शन सी इज करेक्ट. DNA ऑक्सीडेज. DNA ऑक्सीडेज does not take part in DNA replication. Next, which hormone is a modified amino acid? Progesterone, prostaglandins, epinephrine, estrogen. Option C is correct. Which hormone is a modified amino acid? Option C is correct. Epinephrine. एपिनेफ्रिन को और एक नाम से जाना जाता है, वो है एड्रीनेलिन. एड्रीनेलिन से भी जाना जाता है. नेक्स्ट, which amino acid is substituted in sickle cell anemia? ऑप्शन है ग्लूटामिक एसिड बाय बेलाइन इन अल्फा चेन, ग्लूटामिक एसिड बाय बेलाइन इन बीटा चेन, बेलाइन बाय ग्लूटामिक एसिड इन बीटा चेन, बेलाइन बाय ग्लूटामिक एसिड इन अल्फा चेन. ऑप्शन बी इज करेक्ट. Which amino acid is substituted in sickle cell anemia? Option B is correct. Glutamic acid by valine in beta chain. Next, one turn of bDNA double helix spans. One turn of bDNA double helix spans a distance of 3.4 nanometer, 2.46 nanometer, 4.56 nanometer, 4.26 nanometer. Option A is correct, 3.4 nanometer. One turn of bDNA double helix spans a distance of 3.4 nanometer. Next, histone proteins found in the nuclei of eukaryotes are rich in. Histone proteins found in the nuclei of eukaryotes are rich in glycine and phenylalanine, lysine, arginine, glycine, arginine, phenylalanine, and lysine. Option B is correct. Lysine and arginine. Histone proteins found in the nuclei of eukaryotes are rich in lysine and arginine. Next, one of the most common enzyme found in peroxisome is hydrolase, catalase, dehydrogenase, reductase. Option B is correct, catalase. One of the most common enzyme found in peroxisome is catalase. Next, larger subunit of ribosome is composed of. Larger subunit of ribosome is composed of how many RNA molecules? 1, 2, 3, 4. Option B is correct, 2. Larger subunit of ribosome is composed of how many RNA molecules? Option B is correct, 2. Next, DNA synthesis is known as option is conversion, endorsement, rotation, replication. Option D is correct, replication. DNA synthesis is known as replication. Next, a signal peptide is, option A, is most likely to be found on proteins produced by bacterial cell. Option B, direct an mRNA molecules into internal space of the ear. Option C, is a sign to help to bind the small ribosomal unit at the initiation codon. Option D, owned by the first 20 or so amino acid of a protein distinct. Option D is correct. A signal peptide is owned by the first 20 or so amino acid of a protein distinct. Next, the core of nucleosome is made up of following histones. The core of nucleosome is made up of the following histones: H1, H2A, H2B, H3, H1, H2A, H2B, H4, H1, H2A, H2B, H3, H4. Option D is correct, H2A, H2B, H3, H4. Next, lysine is an essential amino acid because it is very rare, it has high nutritive value, it is an important constituent of all proteins, it is not formed in the human body, and has to be provided through diet. Option D is correct. Lysine is an essential amino acid because it is not formed in the human body and has to be provided through diet. Next, the specificity of a protein depends upon. Option A is quantity in which it is present in the system, linear sequence of amino acid in the molecule. Option C, other proteins present in the system. Option D, time of its synthesis. इसका जो करेक्ट ऑप्शन है ऑप्शन बी इज करेक्ट. The specificity of a protein depends upon linear sequence of amino acid in the molecule. Option B is correct. The specificity of a protein depends upon linear sequence of amino acid in the molecule. Next, a nucleotide is thymidine, cytosine, uridylic acid, glutamic acid. Option C is correct. A nucleotide is uridylic acid. Next, which one of the following nucleotide sequence contains 4 pyrimidine bases? 4 pyrimidine bases. इसका जो करेक्ट ऑप्शन है GTGCC 4 pyrimidine bases. Option D is correct. Which one of the following nucleotides contains 4 pyrimidine bases? 2 cytosine है and 2 thymine है, तो ऑप्शन डी इज करेक्ट. नेक्स्ट, deficiency of vitamin K leads to failure of blood clotting, nephritis, anemia, sterility. Option A is correct, failure of blood clotting. Deficiency of vitamin K leads to failure of blood clotting. Next, riboflavin is a vitamin, plant, antibiotic, coloring matter. Option A is correct, riboflavin is a vitamin. विटामिन B2 जो है इसका केमिकल नेम है राइबोफ्लाविन. विटामिन B2 इज राइबोफ्लाविन. नेक्स्ट, आफ्टर व्हिच प्रोसेस डज जेनेटिक प्रोइंसुलिन मैच्योर्स इनटू इंसुलिन? आफ्टर व्हिच प्रोसेस डज जेनेटिक प्रोइंसुलिन मैच्योर्स इनटू इंसुलिन? ऑप्शन इज after addition of C peptide, after removal of C peptide, after removal of disulfide bridge, all of the above. Option B is correct. After which process does genetic proinsulin matures into insulin? Option B is correct. After removal of C peptide. Next, which of the following amino acid is not optically active? कौन सा amino acid जो है optically active नहीं है? Glycine, valine, isoleucine, leucine. Option A is correct, glycine. Glycine is not an optically active. बाकी जो तीनों हैं ये optically active है, सिर्फ glycine is not optically active. Next, restriction enzyme is responsible for which technique? Restriction enzyme is responsible for which technique? Option is respiration, protein synthesis, fat metabolism, genetic engineering. Option D is correct. Restriction enzyme is responsible for genetic engineering technique. Next, what is the catalyst for the decarboxylase enzyme of the cycle? Question 75. What is the catalyst for the decarboxylase enzyme of the cycle? MNCU-FeMO. Option A is correct, manganese. Which is the catalyst for the decarboxylase enzyme of the cycle? Option A is correct, manganese. Next, the central part of the nucleosome is made up of which histone? ये पहले भी एक बार मिला है ये क्वेश्चन, तो इसका जो करेक्ट ऑप्शन है ऑप्शन सी इज करेक्ट, H2A, H2B, H3, H4. The central part of nucleosome is made up of which histone? H2A, H2B, H3, H4. Next, which of the following is a biological scissors? Option is enzyme, restriction endonuclease, restriction exonuclease, D restriction DNase. Option B is correct, restriction endonuclease. Which of the following is a biological scissors? Option B is correct, restriction endonuclease. Next, the core of nucleosome. The core of nucleosome is made up of the following histones. The core of nucleosome is made up of the following histone. Option D is correct, H2A, H2B, H3, H4. H2A, H2B, H3, H4. Next, in amphibia, the egg is of the following type: mesolacetal, macrolacetal, microlacetal, centrolacetal. Option A is correct. In amphibia, the egg is of the mesolacetal type. Next, to prove the one gene one enzyme hypothesis, Biddle and Tatum conducted experiments on which organism? Biddle and Tatum conducted experiments on which organism? Option is E. coli, nostoc, Drosophila, Neurospora. Option D is correct, Neurospora. Next, Lock and key hypothesis was proposed by Koshland, Emil Fisher, Douglas, Willy Kuhne. Correct option is option B, Emil Fisher. Lock and key hypothesis was proposed by Emil Fisher. Next, maltose on hydrolysis with maltase enzyme gives only glucose, galactose, only fructose, only galactose. Option A is correct, only glucose. Maltose and on hydrolysis with maltase enzyme gives only glucose. Next, which of the following is an antimicrobial peptide synthesized by lymphocyte and epithelioid cells? Which of the following is an antimicrobial peptide synthesized by lymphocyte and epithelioid cell? Option E, adhesins, condensins, defensins, cohesins. Option C is correct, defensins. Which of the following is an antimicrobial peptide synthesized by lymphocyte and epithelioid cells? Option C is correct, defensins. Next, cellulose is monosaccharide, polysaccharide, disaccharide, trisaccharides. Cellulose is a polysaccharide. Option B is correct. Next, reagent used to identify protein is question 85. Reagent used to identify protein is Felling's solution, Tollens reagent, Bayer's reagent, concentration HNO3. Option D is correct. Reagent used to identify protein is concentration HNO3. Next, amino acid that does not take part in alpha helix formation is histidine, tyrosine, proline, tryptophan. इसका जो करेक्ट ऑप्शन है ऑप्शन सी इज करेक्ट. Amino acid that does not take part in an alpha helix formation is proline. Next, an amino acid having a hydrophilic side chain, alanine, proline, methionine or serine. Option D is correct, serine. An amino acid having a hydrophilic side chain is serine. Next, a type of proteolytic enzyme is found in infants. Gastric juices, which helps in digestion of milk proteins. The name of the enzyme is peptide renin amylase oxytocin. Option B is correct, renin. A type of proteolytic enzyme is found in infants. Gastric juices, which helps in the digestion of milk proteins. The name of the enzyme is renin. Next, which statement about enzymes is true? Option is enzyme accelerate a reaction by lowering the activation energy. Enzymes are protein which three-dimensional form is key to their function. Option C, enzymes do not alter the overall change in the energy for a reaction. एंजाइम के लिए ए बी सी तीनों करेक्ट है. ऑप्शन डी इज करेक्ट, ऑल ऑफ द अबव. Next, a nucleotide is formed of purine, pyrimidine, phosphate, nitrogen base, sugar and phosphate, purine, sugar, phosphate, pyrimidine, sugar and phosphate. Option B is correct. A nucleotide is formed of nitrogenous base, sugar, phosphate. Next, which sugar is present in RNA? Beta D ribose, beta D fructose, beta D galactose, beta D2 deoxyribose. Option A is correct, beta D ribose. Which sugar is present in RNA? beta D ribose. Next, identify the complementary strand of the DNA primary structure. Question 93. Identify the complementary strand of the DNA primary structure. Primary structure AT GCC GATC. Option B is correct. Identify the complementary strand of the DNA primary structure is complementary structure बोला है TA CG and GC TAG. Option B is correct इसके लिए. Next, distance between two base pair in a DNA. Distance between two base pair in a DNA. Option 0.24 nanometer, 20 nanometer, 0.34 nanometer. Option C is correct. Distance between two base pair in DNA is 0.34 nanometer. Next, which antibiotic inhibits the binding of amino acyl tRNA to ribosome? Which antibiotic inhibits the binding of amino acyl tRNA to ribosome? Tetracycline, penicillin, neomycin, streptomycin. Option A is correct, tetracycline. Which antibiotic inhibits the binding of amino acyl tRNA to ribosome? Option A is correct, tetracycline. Next, long chain of sugar are called polysaccharide, triglyceride, polynucleotides, polypeptides. Option A is correct. Long chain of sugar are called polysaccharide. Next, which of the following contains sulfur? Histidine, proline, serine, methionine. Option D is correct, methionine. Which of the following contains sugar? Methionine. Next, which of the following is aromatic ring? Aromatic ring containing amino acid. Aromatic ring means benzene ring. Which of the following is aromatic ring or benzene ring containing amino acid? Glycine. Option is glycine, tyrosine, alanine, valine. Option B is correct, tyrosine. Tyrosine is a aromatic ring containing amino acid. Next, which one is polyunsaturated fatty acid? Option is glycerol, arachidonic acid, cholesterol, stearic acid. Option B is correct. Arachidonic. Which one is polyunsaturated fatty acid? Option B is correct, arachidonic acid. Next, difference between amino acid are found in. Difference between amino acid are found in amino group, R group, peptide bond, carboxyl group. Option B is correct, R group. R group. Next, which one of the following is universal solvent? Universal solvent नीचे दिया हुआ कौन सा universal solvent है? Option is acetone, oil, water, petrol. Option C is correct, water. Water is a universal solvent. Next, which one of the following bases is found only in RNA and not is DNA? Which one of the following nitrogenous base is found only in RNA? Option C is correct, uracil. Next, the primary structure of protein is due to ionic bond, peptide bond, hydrogen bond, S single bond, S linkages. Option B is correct. The primary structure of protein is due to peptide bond. Question is, the primary structure of protein is due to peptide bonds. Next, which biomolecule fights against infectious organism? Which biomolecule fights against infectious organism? Lipid, nucleotide, protein, enzyme. Option C is correct. Which biomolecule fights against infectious organism? Option C is correct, protein. Next, which of the following is linked together to protein synthesis? Which of the following linked together to protein synthesis? Proteins linked together to proteins. Option A is phosphate, nitrogenous base, sugar, amino acid. Option D is correct, amino acid. Next, chitin is a polysaccharide, nitrogenous polysaccharide, lipoprotein, protein. Option B is correct, chitin is a nitrogenous polysaccharide. Chitin is a nitrogenous polysaccharide. Next, which one of the following is not correctly matched? Triose sugar, glyceraldehyde, aldose sugar, option A. Option B, pentose sugar, ribulose, ketone sugar, hexose sugar, fructose, aldose sugar. Option D. Option D is not correctly. Option C is not correctly matched. फ्रक्टोज जो है ये कीटोन शुगर है. फ्रक्टोज इज ए कीटोन शुगर, नॉट ए एल्डोज शुगर. ऑप्शन सी इज इनकरेक्टली मैच. और ए, बी, डी ऑल आर करेक्टली मैच. नेक्स्ट, which of the following groups is not an organic group? Fats, proteins, enzymes and hormones. Option B is correct. Cofactors and hormones जो है ये all are organic. Cofactors and hormones are organic. Water and minerals are inorganic. Option B is correct. Next, which amino acid acts as a main donor of amino acid in transamination? Which amino acid acts as a main donor of amino group in transamination? Glutamic acid, glutamine, glutamate, dehydrogenase, glycine. Option A is correct, glutamic acid. Which amino acid acts as a main donor of amino group of transamination? Option A is correct, glutamic acid. Next, which one is the most abundant protein in animal world? Collagen, keratin, rubisco, hemoglobin. Option A is correct. Which one is the most abundant protein in the animal world? Option A is correct, collagen. Next, which of the following is not a disaccharide? Maltose, starch, sucrose, lactose. Option B is correct. B is not a disaccharide. Starch जो है ये starch is a polysaccharide. Next, the most common monomer of carbohydrate is glucose, fructose, sucrose, maltose. Option A is correct, glucose. The most common monomer of carbohydrate is glucose. Next, the chemical formula of starch is C6H10O5. Option A is correct, C6H10O5N. Next, Theodiner discovered a free infectious RNA, free infectious DNA, infectious protein, bacteriophage. Option A is correct, free infectious RNA. Theodiner discovered a free infectious RNA. Next, which of the following shows coiled RNA strand and capsomers? Which of the following shows coiled RNA strand and capsomer? Poliovirus, tobacco mosaic virus, measles virus, retrovirus. Option B is correct, tobacco mosaic virus. Which of the following shows coiled RNA strand and capsomers? Option B is correct, tobacco mosaic virus. Next, poliovirus have dsRNA, dsDNA, ssRNA, ssDNA. Option C is correct. poliovirus have single stranded RNA. जितने भी वायरस रहता है उसमें सिंगल स्टैंडर्ड RNA रहता है. नेक्स्ट, व्हाट इज ग्वानिन? ग्वानिन क्या है? व्हाट इज ग्वानिन? प्यूरिन, हार्मोन, एंजाइम, pyrimidine. ग्वानिन इज ए प्यूरिन का जो नाइट्रोजनस बेस है प्यूरिन, एडिनिन, ग्वानिन, डबल रिंग स्ट्रक्चर है. एंड pyrimidine जो है ये साइटोसिन, थाइमिन, यूरासिल, ये सिंगल स्टैंडर्ड रिंग है. नेक्स्ट, द DNA strands are antiparallel. DNA strands are antiparallel because of ester bond, phosphodiester bond, disulfide bond, hydrogen bond. Option B is correct, phosphodiester bond. The DNA strands are antiparallel because of phosphodiester bond. Next, which is the part of DNA molecules? That varies among DNA molecule? Option is pentose sugar, nitrogenous base, phosphate, both A and B. Option B is correct, nitrogenous base. What is the part of DNA molecules that varies among DNA molecule? Option B is correct, nitrogenous base. Next, which one of the following is the major component of chromosome? Major component of chromosome: nucleic acid, protein, nitrogenous base, lipid. Option A is correct, nucleic acid. Nucleic acid. The chromosomes are mainly made up of DNA. Chromosome जो है are mainly made up of DNA. Next, which of the following enzymes will act at pH 1.2? Option lipase, pepsin, maltase, trypsin. Option A is correct, pepsin. Next, protein is synthesized in the free medium by. Question is, protein is synthesized in cell-free medium by Corana, Geiger, Marshall, Nirenberg, Watson. Option C is correct, Marshall, Nirenberg. Protein is synthesized in cell-free medium by Marshall, Nirenberg. Next, the polymerization of RNA. Polymerization of RNA with defined sequences in a template independent manner was done using DNA polymerase, restriction endonucleases, polypeptide phosphorylase, RNA polymerase. Option C is correct. The polymerization of RNA with defined sequences in a template independent manner was done using. Option C is correct, polypeptide phosphorylase. Next and the last question है, the DNA fingerprinting we need. Cells from hair tips, any cells from the body, nucleated cells from the sample. Option D, cells from the ribosome. Option C is correct. For DNA fingerprinting we need nucleated cells from the sample. तो ये था टोटल 127 क्वेश्चन. ये सब प्रीवियस ईयर में आया हुआ क्वेश्चन है डिफरेंट यूनिवर्सिटी का, एंड आप लोग भी सॉल्व करते रहिए. थैंक्स फॉर वाचिंग.